Welcome to DermWire News, powered by DermWire.com. DermWire News for Smart Speaker is made possible with advertising support from Ortho Dermatologics. I'm Stephanie Taleo with Practical Dermatology magazine. A compound extracted from the Christmas berry primrose plant may stop the growth of uveal melanoma, according to preliminary tests. With further testing, the discovery could lead to new therapeutic options for patients with uveal melanoma, report researchers from the Sidney Kimmel Cancer Center, Jefferson Health, and the Icahn School of Medicine at Mount Sinai. I'm very optimistic, says Jeffrey Benovic, Thomas Eakins Endowed Professor of Biochemistry and Molecular Biology at Thomas Jefferson University, and an Associate Director with the SKCC, who led the new work. If the results are confirmed in animal models and eventually humans, it could offer a new way to treat metastatic uveal melanoma patients down the road, he adds. Dr. Benovic and team published their findings in the journal Molecular Cancer Research. The researchers tested whether a compound derived from an ornamental plant in the primrose family, known as Ardisia crenata, might be able to fight the disease. The compound, dubbed FR900359, or simply FR, was discovered 30 years ago from the plant's leaves. FR works by blocking a particular type of G protein that sits on a cell's membrane called GQ, an important signaling molecule. But a subset of these proteins are mutated in uveal melanoma, turning on a molecular pathway that leads to cancer growth. Dominic Lapadula, a graduate student in Dr. Benovic's lab, grew 3 different types of uveal melanoma cells that have the cancer-sparing mutations in the lab. Then he treated the cells with FR. We didn't expect it would work because previous research suggested a related compound called YM254890 did not inhibit the mutated forms of proteins found in uveal melanoma, says Lapadula. But lo and behold, FR very effectively blocked the growth of uveal melanoma cells. When the uveal melanoma cells were treated with small amounts of FR, the cells appeared to revert from cancer cells to typical melanocytes. FR appears to be able to help reset the cells back to their normal state, Dr. Benovic adds. Ideally, that's what you want. Higher doses of FR killed the cells, the researchers report in the new study. The results suggest that the compound could be an effective drug to treat uveal melanoma one day. This study emanated from a team effort within the Cancer Cell Biology and Signaling Program in the SKCC in collaboration with Dr. Julio Aguirre-Hiso at the Icahn School of Medicine at Mount Sinai. Next, Dr. Benovic would like to repeat the findings in a mouse model of uveal melanoma. To do so, he'll team up with Dr. Takami Sato, director of the Metastatic Uveal Melanoma Program at Jefferson. I'm hopeful FR and related compounds will reset the cancer cells in the mouse model, as it did in the cells we grew in the lab, Dr. Benovic says, getting it one step closer to testing in humans. Thank you for listening to DermWire News, powered by DermWire.com. This editorially independent program is supported with advertising from Ortho Dermatologics.